Hello. In today's video we will be examining how temperature affects the rate of anaerobic respiration. For plants and fungi, this process is also known as fermentation. Anaerobic respiration is the process by which energy is produced from food without using oxygen. Like other chemical processes, temperature will affect how fast or slowly the process takes place. You might find the results of this experiment somewhat surprising. You'll need a spirit or Bunsen burner and tripod, a thermometer, 2 flasks, a hold flask stopper, a bowl to act as a water bath, a plastic tube, 200 milliliters of water, 10 milliliters of yeast, and about 100 milliliters of sugar and glucose solution, and a timer or stopwatch. We first make the sugar solution by dissolving a teaspoon of sugar or glucose in water. We then add the yeast to the sugar solution, mixing it well. The sugar is the food from which the yeast microbes will produce energy in anaerobic respiration. Now it's time for the actual experiment. Firstly, we want to place the yeast solution in the water bath. We then close the flask with a stopper, which has a tube running into a separate flask of water. We then light the spirit burner and observe the temperature at 10-degree intervals. Carbon dioxide released by the anaerobic respiration in the yeast goes from the flask through the tube and produces bubbles in the second flask of water. The number of bubbles we see released from the tube will indicate how fast or slow the rate of respiration is taking place. We therefore will measure how many bubbles are produced in 1 minute for every 10 degrees rise in temperature. At first, only 1 bubble was produced per minute at room temperature. At 30 degrees centigrade, that doubled 2 bubbles per minute, and doubled again to 4 bubbles per minute at 40 degrees. The rate of respiration reached its peak at 50 degrees with 5 bubbles released per minute. But at 60 degrees it started to slow down to 3 bubbles until at 70 degrees no bubbles were produced and respiration had stopped completely. This is because at that temperature the enzymes in the yeast had denatured. If an enzyme is denatured, it means that it isn't working because it is dead or inactive. This process of fermentation of yeast produces not only carbon dioxide, but also produces ethanol. That is why it is used in the production of alcohol. Knowing the correct temperature in which to produce the optimum rate of respiration is therefore important for the production of alcohol products such as beer. Other factors that can change a plant or fungi's rate of respiration are carbon dioxide levels, nutrient availability, and amount of sunlight. Thank you for watching.